continuation of genetic code related portion. Next topic, nonsense suppressors also read normal termination signals. The act of nonsense suppression can be viewed as a competition between the suppressor tRNA and the release factor. When a stop codon comes into the ribosomal A site, either read through or polypeptide chain termination will occur depending on which arrives first. Suppression of UAG codons is efficient. In the presence of the suppressor tRNA, more than half of the chain terminating signals are read as specific amino acid codons. E. coli can tolerate this misreading of the UAG stop codon because UAG is used infrequently as a chain terminating codon at the end of open reading frames. In contrast, suppression of the UAA codon usually averages between 1% and 5% and mutant cells producing UAA suppressing tRNA grow poorly. This is expected from the fact that UAA is frequently used as a chain terminating codon and its recognition by a suppressor tRNA would be expected to result in the production of many more apparently long polypeptides. Providing the validity of the genetic code, the code was cracked, as we have seen, by means of biochemical methods involving the use of cell-free systems for carrying out protein synthesis, but molecular biologists are generally suspicious of a method that relies on in vitro analysis alone. So, how do we know definitely that codes are depicted in true living cells? Of course, in the modern era of large-scale DNA sequencing, in which the entire nucleotide sequences of the genomes of diverse organisms ranging from microbes to man have been determined, the genetic code has not only been validated but shown to be universal or nearly so. And thus, a classic and instructive experiment in 1966 helped to validate the genetic code well before DNA sequencing was possible. The experiment was based on the construction by genetic recombination of a mutant gene of phage T4 that harbored a mutually suppressing pair of insertion and deletion mutations similar to the example, and the gene in question encoded a cell wall degrading enzyme called lysozyme, chosen because it is small, easy to purify, and its complete amino acid sequence was known. The experimental strategy was to compare the amino acid sequence of the doubly mutant protein with that of wild type lysozyme, and the amino acid sequence of the mutant NH2, THR, lysine, valine, histamine, leucine, methionine, alanine, lysine, COOH, and wild type were compared. They were found to differ by a stretch of 5 amino acids, highlighted in bold. This observation suggested that the insertion and deletion mutations had scrambled a short stretch of codons in the message of the mutant. Knowing the consequent effect of these scrambled codons on the amino acid sequence of protein imposed important constraints on the nature of the genetic code. Specifically, if the genetic code, as elucidated in biochemical experiments, is valid, then it should be possible to identify a set of codons for the wild type sequence, given that, like when properly aligned and bracketed with the insertion at one end and deletion on the other, would specify the mutant amino acid sequence. Indeed, such a solution exists, which requires a deletion of a nucleotide at the 5' end of the coding sequence and the insertion of a nucleotide at the 3' end. As you can see, the solution verifies several codon assignments and demonstrates that more than one synonymous codon is used to specify the same amino acid in vivo. Lastly and importantly, you should be able to convince yourself from the solution that translation proceeds in the 5' to 3' direction. Next, the code is nearly universal. The results of large-scale sequencing of genomes have largely confirmed the expected universality of the genetic code. The universality of the code has had a huge impact on our understanding of evolution as it made it possible to directly compare protein coding sequences along and among all organisms for which a genome sequence is available. As we shall see, powerful computer programs are available that can search for and identify similarities among predicted coding sequences from a wide range of organisms. The universality of the code also helped it create the field of genetic engineering by making it possible to express cloned copies of genes encoding useful protein products in surrogate host organisms, such as the production of human insulin in bacteria. To understand the conservation nature of the code, consider what might happen if a mutation changed the genetic code. Such a mutation might, for example, alter the sequence of the serine tRNA molecule of the class that corresponds to UCU, causing them to recognize UUU sequences instead. This would be a lethal mutation in haploid cells containing only one gene, directing the production of tRNA for serine would not be inserted into many of its normal positions in proteins. Even if there were more than one gene for tRNA as in a diploid cell, this type of mutation would still be lethal since it would cause the simultaneous replacement of many phenylalanine residues by serine and cell proteins. In view of what we had just said, it was completely unexpected to find that in certain subcellular organisms, the genetic code is in fact slightly different from the standard code. This realization comes during elucidation of the entire DNA sequence of this much amount of base pair human mitochondrial genome, but is observed for mitochondria in yeast, the fruit fly, the higher plants. Sequence of the regions not to specify proteins have revealed the following differences between the standard and mitochondrial genetic codes. UGA is not a stop codon but codes for tryptophan. Hence, the anticodon of mitochondrial tRNA TRP recognizes both UGG and UGA as if obeying the traditional Wobble rules. Then internal methionine is encoded by both AUG and AUA. In mammalian mitochondria, AGA and AGG are not arginine codons, of which there are 6 in the universal code, but specify chain termination. Thus, there are 4 stop codons UAA, UAG, AGA, and AGG in the mammalian mitochondrial code. In fruit fly mitochondria, AGA and AGG are also not arginine codons but specify serine. Perhaps not surprisingly, mitochondrial tRNA are likewise unusual with respect to the rules by which they decode mitochondrial message. Only 22 tRNAs are present in mammalian mitochondria, whereas a minimum of 32 tRNA molecules are required to decode the universal code according to the Wobble rules. Consequently, when an amino acid is specified for 4 codons with the same first and second positions, only a single mitochondrial tRNA is involved. Recall that a minimum of 2 tRNAs would be required by non-mitochondrial systems. Such mitochondrial tRNAs all have in the 5' Wobble position of their anticodons a U residue which is able to engage in pairing with any of the 4 nucleotides in the third codon position. In case where purines in the third position of the codon correspond to different amino acids from pyrimidines in that position, a modified U in the first position of the anticodon of the mitochondrial tRNA restricts Wobble to pairing with the 2 purines only. Exceptions to the universal code are not limited to mitochondria but are also found in several prokaryotic genomes and in the nuclear genomes of certain eukaryotes. The bacteria Mycoplasma capricolum uses UGA as a tryptophan codon rather than a chain termination codon. Likewise, some unicellular protozoa use UAA and UAG, which are stop codons in the universal code, as glutamine codons. Finally, a codon CUG for one amino acid leucine in the universal code has become a codon for another amino acid serine in the yeast candida. We have just seen that variations in the genetic code may occur that are peculiar to certain organelles and organisms, but differences in the code may also come about with the introduction of novel amino acids into protein sequence that serve in fact to expand the code. How these amino acids may arise through natural or unnatural means, and how they become incorporated into proteins to impressive feats of engineering. Then, at expanding the genetic code, as we have seen, 61 of 64 codons in the genetic code specify the 20 amino acids most commonly found in proteins. Some proteins, however, contain unusual amino acids that are not specified by the code. For example, collagen contains the amino acid hydroxyproline, which is created by hydroxylation of proline after it has been incorporated into the polypeptide chain. And some proteins, for example, collagen contains the amino acid hydroxyproline, which is created by hydroxylation of proline after it has been incorporated into the polypeptide chain. And some proteins contain selenocysteine, which is generated on a specialized tRNA that is charged with serine. The serine is converted to selenocysteine by enzymatic incorporation of selenium. The specialized selenocysteine charged tRNA enters the ribosome. The elongation factor introduces the selenocysteine charged tRNA into a site where the metal containing amino acid is incorporated into the growing polypeptide chain. These and other cases of modified amino acids involved specialized mechanisms for introducing altered amino acids into proteins without violating the near universality of genetic code. But what if through genetic engineering we could expand the genetic code to specify unnatural amino acids? Could we do so in a manner that allows us to incorporate tailor-made amino acids at a particular sites in proteins and thereby generate novel proteins and even whole organisms with useful properties? The convergence of two lines of research has brought these possibilities into the realm of reality. In one, the tRNA has been created that recognizes UAG, but that is not a substrate for any of the existing amino acid tRNA synthetases in E. coli. Instead, cognate synthetases have been generated by an iterative evaluation strategy that recognize the unique tRNA and recharge it with a particular artificial amino acid. In this way, a series of synthetases have been evolved that recognize and charge the cognate tRNA with one of a variety of unnatural amino acids with usual features such as heavy metal binding sites for facilitating X-ray differentiation studies, fluorescent moieties for fluorescence microscopy, photo cross-linking sites, or chemically reacting groups. E. coli strains producing the UAG recognizing tRNA and one of these novel synthetases are capable of taking up a cognate unnatural amino acid from the growth medium and incorporating it into proteins at UAG sites in the mRNA. For example, if we want to introduce a photo cross-linking site at a particular position in a protein of interest, we introduce a tag codon into the coding sequence for that protein and express the tag containing gene in E. coli cells engineered to introduce the unnatural amino acid at UAG codons. Meanwhile, in a second line of research, an E. coli strain is being created in which all 314 tag UAG stop codons in the E. coli genome are being replaced with TAA or UAA stop codons. This engineering feat is achieved in two stages. In the first stage, a multiple approach known as MAGE, multiplex automated genome engineering, has been used to create 32 strains in which different subsets of TAG codons have been replaced. Then different examples of unnatural amino acids developed for incorporation by an expanded genetic code is there. Now we are going to learn about a quick summary of this topic. Before that, continue this. Like with synonyms, stop codon, next strategy based on hierarchical genetic conjugation known as CAGE, conjugative assembly genome engineering, is being employed to merge all 314 codon stimulations substitutions into a single strain. This strategy promotes and promises to culminate the E. coli strain in which the codon TAG has been freed up for the incorporation of tailor-made amino acids. In the future, strategies such as this could be applied to other microorganisms such as yeast. Unshackled from the constraint of 20 natural amino acids, E. coli and yeast engineered to have a 21st amino acid specific codon might have a greater capacity to evolve usual rates under controlled laboratory conditions than their uncontrolled and unmodified ancestors. In the universal genetic code used by every organism from bacteria to humans, 61 codons signify specific amino acids and remaining 3 are chain termination codons. The code is highly degenerate with several codons or synonyms usually corresponding to a single amino acid. A given tRNA can sometimes specifically recognize several codons. This ability arises from movable in the base at the 5' end of the anticodon. The stop codons UAA, UAG, and UGA are read by specific proteins, not specialized tRNA molecules. The genetic code is subject to 3 principal rules. Codons are read in a 5' to 3' direction. Codons are non-overlapping and the message contains no gaps. And the message is translated in a fixed reading frame which is set by the initiation codon. The genetic code was cracked through the study of protein synthesis in cell-free extracts. Addition of new mRNA to an extract depleted of its original messenger component results in the production of new protein whose amino acid sequence are determined by the externally added mRNA. The first and probably most important step in cracking the genetic code occurred when the synthetic polyribonucleotide poly-U was found to code specifically for polyphenylalanine. Use of other synthetic polyribonucleotides, both homogeneous poly-C and so on and mixed poly-AU and so on, then allowed assignment of codons for the various amino acids. Determination of the exact order of nucleotides and codons subsequently came from a study of specific trinucleotides, tRNA ribosome interactions, and the use of regular copolymers as messengers. Point mutations that alter the code as missense mutations, which change the codon for one amino acid into the codon for another amino acid. Nonsense mutations, which cause protein synthesis to terminate prematurely, and frame shift mutations, which alter the reading frame of the message. In some cases, the effects of missense, nonsense, and frame shift mutations can be partially suppressed by extragenic suppressors. For example, mutant tRNAs can stop codons generated by nonsense mutations as if they were codons for a specific amino acid. A slightly different genetic code is utilized in mitochondria and in the principal genomes of certain prokaryotes and protozoa, such as the use of UGA, a stop codon in the universal code as a tryptophan codon.